Okay, well, it's great to be back in the piece. I started up here in 2008. So without the Peace Region Forage Seed Association, I wouldn't be where I am today. So thank you. So today, my research has shifted a bit, and I'm going to talk about alfalfa weevil. Previously, I worked on red clover up here. You all know this, of course. Canadian prairies were the heartland growing area, breadbasket of Canada with over 76 million acres. When we talk about alfalfa seed, it's grown throughout the prairies. In Alberta, mainly is, whoops, sorry, that's not a pointer. As pointed out earlier today, it's mainly southern Alberta, and then Saskatchewan, we have various regions, quite a bit in the northeast, and then Manitoba, primarily that interlaken region. And I heard there was anywhere from 60 to 80,000 acres in production for alfalfa seed, both certified and common. If we think about Alberta, I know if you looked at Nicole's poster earlier, she would say, you know, Oregon is the world's forage seed capital. Well, Alberta, I would consider Canada's forage seed capital, with most of that, of course, being up here. A lot of the grasses and other clovers and whatnot up here in the piece, and then alfalfa primarily in the south, and particularly certified alfalfa seed. So going after this seed, unfortunately, it's not only us trying to harvest it, but a weevil trying to eat it. And so here's that little beast, the alfalfa weevil. Most of you probably know this, but there's one generation per year. It overwinters as an adult. In the spring, it emerges. They mate. The females will chew a little groove in the stems and lay eggs. The larvae will then hatch, and they'll move up and feed on the developing buds and terminal foliage. They'll go through 4 instars, meanwhile getting bigger and bigger, and eventually, under high pest pressure, can lead to significant defoliation of the crop. Now, if we think about alfalfa and alfalfa weevil, neither are native species to North America. Alfalfa weevil was first found in southern Alberta and Saskatchewan in the late or probably mid 50s. It really stayed around there and kind of below. Oh, I don't have it on this slide. Below like the 51st parallel, before moving further east into Manitoba, and then further north up into the Melfort-Tisdale region. So this is where we're currently at, although I should put a star. It's also found in BC as well, now just within the past few years. Luckily for us, it's not up in the piece yet, and hopefully it doesn't get up here. But really, starting, depending on who you talk to, anywhere 2008 to 2015, we started having issues with alfalfa weevil. And particularly here in Alberta, by 2015, we were having really ineffective control with some of the synthetic pyrethroids, so your Matador or your Decis for weevils. And around 2018, I was a research scientist at the time with AFC in Saskatoon, and working with Hector Carcamo, we did some sampling in Rosemary, a couple fields up there, and a site down near Lethbridge. The Lethbridge site was an alfalfa site on AFC land that no insecticides had been applied to, whereas these 2 other fields were commercial certified seed production fields. So we went out to these sites and we collected them. We had suspected resistance from our seed production sites and not from our site near Lethbridge. We did a very standard resistance test, so there's standardized methods for insecticide resistance testing. We call it a vial bioassay. And so for this, when we first started, we were just using the commercial product, so this would have been Decis at the full recommended rate, and then we halved it, quarter, and a control. And you can put your solution in your vial, you rotate the vial, and it coats the interior of the surface. Then once the interior of the surface is coated, you can add your weevils. You come back 24 hours later, and you can score them, whether they're dead or alive. And when we did this is what we found. So immediately, probably what jumps out, First off, nothing died in the control. That's good. But then, as you probably noticed, Lethbridge, we had very effective control, even at a quarter rate. But then you'll notice the 2 rosemary sites, ineffective control, about 50% at most. And if we're talking about commercial products, we want to hope for at least, I would say, 90% efficacy, and anything below 90, then we get into kind of suppression rather than control, and then probably below about 70%, then you're looking at some form of resistance. And then we did this. We went out to other sites. Now we're jumping up a few years. We did other work in the meantime, but I'm just going to gloss over it. But now we jumped to 2022, and we did a survey, and we just used 1 dose, the full recommended rate, across several. All of these are commercial production fields. And unfortunately, the highest mortality we found during 2022 was about 50% mortality. So again, showing that no matter the field, we were having resistance within our weevils. So what do we do now? Well, we can think about some alternative tactics, and one is the potential for biological control. So as Lindsay was just talking about parasitoids, well, here's Oomysis incertus. This is a parasitoid of the alfalfa weevil, tiny little one. There's also another species. This is Bathyplectes curculianus, another parasitoid of our weevils. But it's really difficult to identify a parasitized weevil. You can't just look at it and say, oh, that one looks sick or unhealthy. And so you can either rear them in the lab, and we did that. One of my former students, Michelle Reed, did this work. And so she would rear individual larvae in these little cups, and she would feed them daily, make sure they were nice and happy. We were aiming for at least 200 individuals per field, and then we'd stack them up in the growth chamber, growth room like this. But this takes a lot of time and effort. You got to make sure you have a nice fresh source of alfalfa close by. So we were also looking at other ways to identify parasitized larvae. And so we developed a molecular method. We've heard a lot about molecular methods today. This one's fairly simple compared to some of the other ones we've heard about. So what we can do is we can identify these based off of species-specific segments of DNA. And so to do that, we've sequenced the DNA, particularly the ITS region. Doesn't really matter what region, but we have different primer sets, and so each primer set will amplify a fragment of DNA that's a different size. And so based off of the size of that fragment of DNA, we can determine what species it is. And so with this, all we have to do is take our weevil larvae, grind it up, extract the DNA, put in our primers, run a reaction, run that on a gel, and then this gel DNA, we load it at the top. DNA separates out by size, so larger DNA fragments are at the top, smaller DNA fragments are at the bottom. And then because we have 3 different species, we will have potentially 3 different bands on our gel. And so this is what it looks like. These are controls, we call them ladders of known size standard DNA. Then we have our alfalfa weevil, our Bathyplectes, and our Oomysis. And so the Bathyplectes fragment is a little bit bigger, the Oomysis is a little bit smaller, and then alfalfa weevil is in the middle, and that kind of acts as a positive control to make sure it's alfalfa weevil, and it makes also sure that your reaction works. And so looking at this gel, then we can see, okay, with these top bands, all of these individuals would be parasitized by Bathyplectes, and all of these individuals below would be parasitized by Oomysis incertus. And this is helpful. Michelle went out and collected from a bunch of commercial fields, and then looked at compared rearing, so in black, this is the live rearing in the laboratory versus our DNA molecular method. And this is pretty typical for molecular methods. She found higher parasitism levels using the molecular method compared to rearing. And it was a bit variable across different fields, but the nice thing is we were seeing fairly high parasitism levels across some of those fields. And most of the time, our molecular method was picking up the parasitoids more so than by rearing. Interesting things you can do with this is you can collect the weevil larvae each week, and then you can look at the kind of the seasonal phenology of the weevil and when the parasitoids might be starting to be present within the weevils. And so this is just looking at total percent parasitism across all the samples. The black bar is the average, and then the other ones are for individual sites. There's going to be variability, of course, because you're sampling each week new individuals. But we've separated it out here. So this was parasitism by Bathyplectes, parasitism by Oomysis, and we did occasionally find some weevil larvae that were parasitized by both species. We don't yet know, and this is where we're trying to go forward in the future, is what parasitism levels do we need to really start having some effective management of our alfalfa weevil populations. If you're familiar with Julie Soroka, Jennifer Otani, some of their work, they always have this: if you have 3 parasitoids present in your region, you generally don't have issues with alfalfa weevil. Unfortunately, we only have 2. A third one's been found occasionally, but not very often. So with our 2, what kind of levels do we need to get to have some effective management by them? So this was a study. Michelle's work was funded through Canadian Agricultural Partnership Project that was led by the Peace Region Forage Seed Association and also funded by the Alberta Alfalfa Seed Commission. So that ended in 2023, and then since then we want to build on this project. And Jen mentioned this work in her talk yesterday. But where do we go from here? So we know we have insecticide resistance. We have this molecular tool that can help us identify parasitism of our weevils, and so we want to build from here. And this is part of a new project that a master's student, Adele, who's sitting at the back, is leading, where we're reevaluating, or I should say maybe more reinvestigating, the economic thresholds in the context of insecticide resistance and this increased alfalfa weevil pressure that we're seeing in the field. That's not the official name of the project, but we have 4 main objectives. The first major objective is to actually, so we have this fairly effective molecular tool, but you need a research lab. It'll take a few days to run it on, maybe you could do 48 individuals in 2 days. So it can take quite a bit of time to run it. So we're working with Tim Dumonceau, who's a research scientist in AFC Saskatoon, to develop new tools that we can use and potentially be able to identify if a larvae is parasitized in 40 minutes or less. So that's one aspect of this project. We're also developing an on-farm resistance test kit. So farmers themselves, before they go out and spray, they could kind of have an idea of what might be effective against their weevils. We are looking to reassess the current alfalfa weevil thresholds, and I'll talk a little bit more about that today. And then, again, get after this parasitism aspect. What kind of levels of parasitism are we seeing in our fields, and what do we think we might need to get to actually have some more control via parasitoids? So just talking a little bit about some of the work we've done in 2023, where unfortunately bugs were bad in 2024, and we're still processing samples that we collected from 2024 because there's so many insects to identify. But so in 2023, we had 10 field sites, all commercial fields. Oh yeah, sorry, that shouldn't say 2025, but it actually should say 2024. But just to say, the other data that I showed you was from 2022. 2024, this past summer, we're still seeing high levels of resistance. We had one field where they were doing a bit better. That grower was doing a few different things in his field to try and control weevils, and seems to have less resistance issues in that one field. But otherwise, it still seems to be pretty consistent across southern Alberta. Things haven't changed much in a couple of years. But going back to this large-scale field trial, so at these fields, within each field, we had 4 or 8 plots. We basically had 4, we call these our managed strips, which of course would be sprayed. And then we had 4 unsprayed strips, which went along the access to the bee shelters. These were about 300 by 100 by 300 feet. And if you look closely, you can kind of see them in this drone shot. Unfortunately, because they're a little bit lighter in color, those were our unsprayed sites. There's a 4th one, but it's just not in the photo. So looking at 2023 data, this is over several different sites, but it's kind of grouped by closest town/area. And so what we found, this is the number of alfalfa weevil per 10 sweeps, and you can see the seasonal phenology across all of the sites. In the Enchant region, unfortunately, that's where weevil numbers are quite a bit higher. The dashed line would indicate what the lower end of the threshold is. So currently, the threshold is 20 to 30 late instar larvae per sweep. And so that dashed line would be what the threshold was. So if you look at it, there aren't actually, if you're going by the threshold, there aren't actually that many sites that went over threshold. And when we look, this is broken up by, we sampled heavily pre-spray, so basically a few days before the grower would go out and spray, and then after the week after, we'd come back and we'd sample again. And looking at the larvae per sweep, doesn't show up and it's quite small. I apologize, but let's just focus on these bigger bars. So this first bar would be the control prior to spray. The next bar would be the treatment prior to spray. So they should be basically the same, right? Because nothing's been sprayed yet. And then, interestingly, once after the spray, even in the control, we saw a reduced number of weevils. And then in looking at the actual sprayed plot, we see a reduction in weevils. But not a huge reduction, and all of these fields were sprayed. And you can see this is the number of larvae per sweep. Our threshold is up here. So again, if we're thinking about using the actual threshold, none of these fields would have been at threshold. Well, this one field would have been at threshold prior to being sprayed. But I'm going to skip this. I'll come back to those. If we look at yield was still heavily impacted. So on average, this is a box and whisker plot, so it's not yield, it's not means, but you can see there's significant differences in sprayed versus unsprayed plots. And again, these plots were sprayed before the threshold was reached, and there still was significant reduction in yield, in our unsprayed plots. And so if we looked at the average across all of our plots, we had about 800 pounds an acre in sprayed plots and about 400 pounds an acre in our unsprayed plots. So pretty significant reduction, and that's not even really considering the thresholds. So do our current thresholds work? Most growers would probably tell you no. And so we really need to kind of tease this out, and what can we do, where can we get, what kind of numbers do we need for this? So that's 2023. We repeated it last year in 2024. We have the yield data. I'm not going to show that today. We're still processing insect samples, so we're going to redo it one more time in 2025. So we'll have 3 full years of data. We lost a couple sites this past summer. I think we ended up with 7 or 8 sites. So far we have about 17 sites, and we'll aim for another 10 this coming year. What other things, though, we can get from this data is we can look at other insects that we're collecting in our sweep nets. So here we're just looking at the actual sweep net samples for this parasitoid wasp, the Bathyplectes species, and we are picking it up once in a while in some of these sites. We're also currently processing the weevils that we collected with our molecular aspect and to look at the parasitism rates within, and we're going to be comparing that between our sprayed and our unsprayed sites. With sweep samples, we really don't pick up any real big differences, whether or not the plot was sprayed or unsprayed in terms of parasitism. but we're wondering about the actual weevils that we're finding in these plots, what they're like in terms of parasitism. So will there be differences in the actual weevils than in the adult parasitoids that we're finding? Other things we can look at are there's other natural enemies out there. There's predators that will potentially feed on alfalfa weevil. So we can look at, well, we have parasitoids as well, but we can look at ladybugs, other flies that are predators, damsel bugs, lacewings, minute pirate bugs. And so this is going to be built into hopefully a new project where a PhD student, Leo, is going to start looking at these predators and seeing, will they feed on alfalfa weevil? Can we find them actually feeding on alfalfa weevil in the field? And similarly to the parasitism work where we're using molecular tools, we can also use molecular tools to identify what predators are consuming in the field as well. So it's one thing to show in the laboratory that you can feed a beetle alfalfa weevil larvae and it will eat it, but can you actually show that from the field, what it's doing in the field? And so that's part of where we're going with this current project. So Adele is currently reevaluating or reinvestigating the economic thresholds and continuing to do resistance testing. This coming year, we're also going to be doing some testing in Saskatchewan and in Manitoba. When I was working in Saskatchewan, talking to some growers, that would have been 2017, 2018. They were saying they hadn't had effective control with pyrethroids for years, and they were using other products. So we suspect there's resistance in Saskatchewan. We suspect there's probably resistance in Manitoba. And it's not a new thing for alfalfa weevil. If you look in other parts of the world, throughout the US, where Lindsay was showing her map, where that alfalfa was being produced, most of those regions have issues with some resistance to some products. So this wouldn't be this isn't just a made in Alberta thing. So I mentioned the predator aspect. So we're starting to look at the predators and what they're feeding on, and specifically looking at whether they're feeding on alfalfa weevil. Leo, a new PhD student in my lab, will be doing this. And then hopefully together with Bill, we're going to start looking at some germplasm. Because really, where do we go from here? We don't have very many management options for weevil control. So maybe it's time to start looking at the actual host plant. And if there's any traits we can identify in host plants, that then, of course, the plant would be naturally resistant to weevils. I know we're thinking about the seed side of things. So with this work, we'll also have to consider, of course, like the protein content and all of that aspect in terms of the forage quality. But at least we're going to get started down this road because unfortunately, as we've all heard before, there's no silver bullet for this. And so the more different aspects we can throw at this weevil, the better, in my opinion. So with that, I have no idea what I am doing for time. But I just want to thank all of you for being here. Thank you for our funders. Our current project is funded through ARDAR and the Alfalfa Seed Commission. Our previous project was funded through a Canadian agricultural partnership led by the Peace Region Forage Seed Association with Ag Canada and Saskatchewan, as well as the Alfalfa Seed Commission on board. And my current position is also supported by the major crop commissions here in Alberta. So thanks to them as well. And I'd be happy to take any questions. So thanks. If the alfalfa weevil has any secondary hosts? That I don't know. Weldon, would you know the answer to that? I don't. I think it's fairly specific. Yeah. So we still have to dig into this data because like this past summer, we were having aphids in some of our sites, and you could clearly see the unsprayed plots had fewer aphids than the sprayed plots and more predators. So unfortunately, with aphids, sweep nets aren't the greatest way to collect aphids, nor count the thousands that you literally collect. So we've kind of just been saying whether or not aphids have been present or absent. And so that's something we're going to have to dig into is, look at all of these. I was bashing my head against my computer screen yesterday trying to also look at our data. It's a great comprehensive data set but the fields are kind of managed differently, as you know, and so trying to look at, maybe we have to look at time of first spray, time of second spray, and try and tease that apart, what's going to kind of give us the best relationship. When I graphed, I didn't show it today, but when I was graphing just total larvae by yield, there is no relationship. Like, it's all over the map, in sprayed or unsprayed. Sure, unsprayed sites are lower, but it doesn't necessarily mean they had more weevils. So, yeah, exactly. So the threshold that is in the crop production guide is based off of seed crop. So for forages, you're most likely not going to spray an insecticide. If it's really bad, you're probably going to maybe mow, maybe hay earlier. But so yes, we're thinking specifically in a seed crop that we have to look at this threshold. We also did there's other ways. If you look in the US, they have other ways to estimate thresholds. They have a shake bucket, kind of counting stem tips and then shaking how many weevil larvae you have on 30 stems. We've done that as well. I didn't show that data, but it's another kind of way that you can estimate the threshold, and we're going to see, is that a better way to estimate threshold than a sweep net? We don't know, but we're gonna look into that as well. So. There's still 3 years left on this current project. Yeah, Shelby. That would be the ultimate goal, right? Is can we determine, and part of the aspect of the project looking at kind of we already have this molecular tool for parasitism, but it takes a while. So if we can get a quicker parasitism test, can we look and say, okay, you just collected 100 weevil larvae through this test. Before you've sprayed, through this test, 90% of them are parasitized. That would be ideal, right? So then, again, those weevil larvae, they're parasitized. They're going to continue to eat that year. But if we think it's a perennial, over time, maybe we can start building up these parasitoid populations if we hold off on our sprays, or maybe we spray different products, depending on what product we use. And maybe that will help us build up these parasitoids, build up the predators in our fields. And then ideally, yes, we would like to say, okay, this is your threshold, but you can estimate if you have 50% parasitism, then your threshold goes up here. And so those are called dynamic action thresholds. There's only been a few that have been developed. Tyler Wiss developed one for cereal aphids. He's with Ag Canada in Saskatoon. There's been one that's also been developed for soybean aphid, and a couple others in the works. But yeah, that would be the ultimate goal, is trying to factor in the predators and the parasitoids and their impact on these weevils as well. Yeah. All right, great. Thank you very much.